Perfect.
Off to that bad start. Thanks again, and Roxandra. It's me who's honored to be here. I never imagined giving a keynote talk at this stage in this particular meeting. I used to go to these meetings a long time ago, and I kind of went adrift, as you'll hear, from network biology in general. But in fact, it's good timing because I'm coming back to network biology. In fact, I'm not going to talk about any of it today because it's really not quite ready. But it's extremely exciting, and I'm looking forward to getting these stories out. So as I said, it was a long time ago. In fact, it was so long ago that I was at a network meeting that what I recall is hanging out with Marion Wallout and Mark Vidal smoking cigarettes during breaks and during drinks, which I understand none of us do anymore, fortunately. In any case, I also want to thank the organizers for an extremely exciting meeting. I mean, I too am extremely inspired by what's been presented here, and I commend Anne and Pascal and Michael and Roded for putting together this marvelous program, and for leading such interesting discussions. So what I'm going to do today, I've kept the same title, but it's a little different, and the reason was that this meeting was also somewhat harrowing in the sense that I found myself after every session thinking about what I could put into my talk that might have something to do with it. And so things got really schizophrenic. And it wasn't until today, in fact, that I finally formulated the final presentation. So I'll start with some philosophical thoughts about and what I mean by the dimensions of function. So the inspiration for the title was, in fact, from this paper. When I heard from Anne Roxandra, I immediately asked myself, well, what's she been up to recently, and looked up. And I found this interesting philosophical work on the meaning of function. And what she does is goes through very clearly and systematically describing what are and why are there different concepts of what function is, and why it can be a problem, but also how consensi can be found in our understanding of this. And in fact, the discussion of function is one way for us to get clearer ideas of what we're working on. So I think, so at any rate, I want to talk a bit about that. And so I've broken things up into two talks, in fact, or an act in two part. One is what I call a spectroscopist perspective on function, which is something of the way that I came to understand the network biology, how network biology defines function. And I'll talk about some sort of historical background to how I came to these ideas and where we are today. And then I'll talk about this business of biomolecular condensates and why they are kind of breaking the rules of everything that we know about molecular organization, but also at the same time presenting interesting challenges for network biology. And I'll talk about a specific example that we've been working on in the lab involving looking at the role of condensates in endocytosis. So with that, as I said, we'll start with some light philosophy. And I guess if you ask your basic student what network biology is, they might say, if a clever one might say, well, I mean, what they do is they seem to collect a lot of components, molecular components of cells, and they look at interactions and dynamics of these things with each other. And somehow, if they work with some very smart mathematicians and informaticists, they'll be able to perform some sort of mathematical transformations that will bring out some perfect gestalt that tells you why the matter is organized the way that it is. In a more prosaic sense, what we think of, or what I think of as network biology, is understanding how molecular networks go from one state to another, in which the edges and nodes increase and decrease in some sort of quantity, and they do that in response to both the environmental inputs they're getting, as well as the intrinsic effects of the genetic background and epigenetic actions on the network. So what we want to do, of course, is understand these networks, or surrogates for the underlying molecular networks that make up living cells, that make up tissues, ultimately, perhaps someday, what forms human consciousness. And so when I started thinking about this over 20 years ago, to begin with, as and Roxandra mentioned, I started out a physical biochemist, and specifically, one of the techniques I used was proton multi-dimensional NMR. And so just as a quick summary of this, I can explain how the notion of spectroscopy is really akin to the types of notions and problems that we face as network biologists. So let me explain. So I think you all know more or less at least the thumbnail of this process, which is what this is, a thumbnail, in which you have Nmr Spectra. What these peaks here represent is that there are resonant frequencies for the magnetic moments of individual atoms in a molecule. Let's say a protein. So you have nitrogen, carbon, and protons. This is a proton spectrum as it happens. And what this is telling, what these peaks here tell us, they're symmetrical, is that there's an atom that has a nucleus that resonates at this frequency that's coupled to an atom that resonates at this frequency. And what you can get from that information and from the positions of the resonance frequencies and the patterns of cross peaks to different molecules is the identities of the individual atoms in an entire molecule, like a protein. And in addition, there's relaxation processes and other electronic coupling processes that lead to information about the angular and distance interactions between the atoms that ultimately can be thrown into a program, and using the theory of the chemical bond result in some gestalt, which in this case is a protein structure. In addition to that, you can obtain novel information about the dynamics of these molecules and how that affects their function. Okay. So there's a couple of analogies to what we do. First is the notion of a comprehensive list of facts, okay, which are the component atoms of the molecules and their relationships. And this is described very well by the early presentations by Mike Calderwood and Ben Wheeler on both comprehensive and comparative analyses of protein interaction networks that they've reported recently, and the importance of this approach to both provide comprehensive information as well as permit using simpler and more compact approaches for faster types of ways of doing this, such as the sort of interactome in a tube approaches that have been developed by people like Fritz Roth and by Sasha Levy. And so these are the basic elements that you get out of this. So what's happening here? What you have, just to summarize, you have something that you don't care about, which is a probe, which is the nuclei of these, or the nuclear magnetic moment specifically, of all these atoms in this molecule. And what you're interested in, though, are how are these nuclear magnetic moments coupled to each other, and what does that represent about the gestalt, which are the structural elements, in this case amino acids of the molecule, and finally the global organization of that molecule and its dynamics, which we obtained by probing with, in this case, electromagnetic pulses, each and every atom of the molecule. Okay, so here's the question I asked myself over 25 years ago: how do we make a molecular spectrometer of the cell? We need something that we're going to probe, which is a surrogate. It doesn't tell you everything, but it can be a surrogate for other information about molecular information, right? In my case, it seemed to me that the most important simple thing that you could measure would be protein-protein interactions. But ultimately, what you want to be able to do to derive from this information is to derive both structure and dynamics that are going to tell you both about the organization and the directions of flows of information, matter, and energy through a network. I should say, just parenthetically, that one of the challenges, I think, in network biology is that we've heard about all sorts of interesting approaches to mapping the mapping the dynamics of networks using network propagation approaches. For instance, certainly from Roded, and Dana Silverbush gave an excellent talk on this, several days ago. And what those things do seem to be extremely informative, but what they're missing, I think, is the details of the transfer functions between nodes, both in time and dose. And this is something that, I think hopefully more and more as we develop techniques that can give us information about this, we'll learn interesting things. I think Steve Smith gave a great talk showing his particular approach to this problem. So anyway, that led to motivation for something that really came from It was actually, I was at a lecture at MIT by Alex Vershavsky, and he, at the end of his talk, which was all about, of course, ubiquitin protein processing, he threw in this little extra thing about something he was doing called the ubiquitin split protein sensor. I walked out of that talk completely changed. I think I walked around the Charles Basin around about 10 times thinking about what I could do by generalizing this approach. And basically, that's what I've been doing ever since. So the idea, of course, is to harness the unique unidimensional process of protein folding as a tool for detection of protein-protein interactions by dissecting a reporter protein, what will be a reporter protein, into two complementary N and C terminal fragments and fusing them to two proteins of interest. The reasoning being that if the two proteins interact, then it recreates the unimolecular conditions for protein folding that result in reconstitution of the structure and an activity that you can measure. Okay? So this approach it's simple, it's direct. They have other features that we've been able to take advantage of, including being able to do these in practically any cellular context or in vivo that you would want, and be able to obtain spatial temporal information about protein complexes, but at the same time, keeping it relatively simple and scalable. And years have gone by. We've developed a whole bunch of different methods for both spatial temporal and spatial temporal measurements of protein-protein interactions, as well as applications both in vivo and at a large scale, and for even some practical problems like doing network-based discovery of drug actions and mechanisms. So, and you heard about some other examples that have been Leonard gave an excellent talk this morning, of course, where he mentioned using this approach in studying protein evolution, and there's a lot of applications now in synthetic biology. So anyway, one of our ultimate aims, of course, as I mentioned, was to obtain structure, and we were able to demonstrate that you could determine, because of the folding nature of this approach, one obtains both distance information and topology information based on the fact that this folding process requires a specific topology in order to happen, and therefore it gives you information about distances between molecules. And this, of course, for instance, is a contact map showing the interactions that we detected for the nuclear pore complex of yeast, that you can see in the pattern that there's obviously structural information, both direct and indirect, determined by basically a constraint, which is the length of the linkers that link your bait and prey proteins to the fragments. In this case, 8 nanometers resolution. We like to refer to PCA in general as a non-optical super resolution technique. Anyway, so that's great. Then we did this, and of course, at the time, we were thinking everything in the cell was going to look like something like this, right? And in fact, what we got was really, really confounding information that didn't make any sense, okay? And so here's an example that just drove me nuts, trying to figure out what possible organization of matter could this represent. Now, this leads me into a complete segue into my next subject, which is my talk about what I call mechanoactive protein condensates that drive actin-independent endocytosis and perhaps other processes. So how we got into this it started actually with a paper, the story we had here, which was, as Anne mentioned, we had some interest in noise in phosphoprotein networks, and this was an attempt to look at the effects of the importance of or prediction of noise from temporal information about phosphoproteomics. So here are some examples, for instance, of these high temporal resolution traces of phosphorylation of differential phosphorylation of specific sites on proteins as a function of two different states where the cells were osmotically shocked by salt versus a control. Yeah. So what was interesting about these two proteins is, in fact, they belong to a bunch of proteins that just showed up as a cluster in our search, and they happen to be proteins, or so-called proteins, involved in endocytosis. So in yeast, where we did this study, endocytosis is a a temporally defined process in which various proteins. First, you start with clathrin, of course, and then there are adapter proteins and then coat proteins that are recruited that ultimately lead to assembly of the actin polymerization nucleation machinery. And it's the formation of the branch actin that drives membrane invagination into the cell. Now, just as an introduction, not an aside, the important thing about this is that this is how it happens in yeast, but in fact, in most places, actin is not involved. In fact, the sources of energy driving invagination remain largely theoretical and controversial. And and in fact, we can create conditions in yeast in which actin-independent endocytosis becomes the dominant form of endocytosis. Just to show you some of this temporal behavior, it's really quite marvelous to behold. So you have these puncta here in which an individual reporter protein, SLA1, is one of the coat proteins is labeled with GFP, and another protein, ABP1, labeled with RFP. And what you can see is these are chymographs with 30-second interval represented here, which you can see the recruitment of SLA1 for a period of time, and then later of ABP1, which is involved in a much later process. And we overlap them. We can see this. You can see the end of the process by basically the puncta disappearing, going out of the focal plane and disappearing into the cell. All right, so what is this all about? Oh, and by the way, when you shock the cells with NaCl, all of this stops. It just freezes. It's quite remarkable. So another thing that was remarkable is that when we lined up all these proteins that were undergoing large amounts of phosphorylation or dephosphorylation, it turned out that they all had one thing in common, what are called Prion-Like domains. So these are domains that are defined, they were actually defined experimentally by Simon Alberti when he was in Susan Lindquist's lab, but extensively looked at proteins and determined what were the sequence requirements and their compositions that seemed to make them prionic, make them form, prionic bodies. And it turned out that dominant features were things like glutamine to asparagine composition and positional aromatic amino acids, periodic aromatic amino acids, and some other things. So basically, they came up with these sort of rules that define PRDs. Now, these are intrinsically disordered domains entirely, and of low amino acid composition. So there was no sort of simple rule for this, as I'll get into, defining what is one prion domain versus another. And that turns out to be an interesting and challenging problem. So this segues into another thing, which is that there's another thing about prion-like domains that has emerged as very exciting in the last several years. The fact that they organize in a way that was a surprise. So I take you back now, just to take a little side trip into the discovery of biomolecular condensates, and it's an interesting illustration of cross-disciplinary conceptions of organization of matter that I thought would be useful. So the author of this paper, Cliff Brangwyn, was a postdoc in Tony Hyman's lab. And when he arrived in Tony Hyman's lab, Tony asked him to figure out what these things are, what these green dots here are. What these green dots are what are called P granules. They are these ribonuclear protein bodies that polarize to one end of C. elegans embryos and are critical to the polar expression of key genes involved in defining the dorsal ventricle axis of this organism. At any rate, so what did he do? Well, I've got to give you a little background on Cliff. So Cliff did his PhD with a guy named Dave Weiss at Harvard, and some of you will know, particularly single cell people might know of him, particularly those in the Boston, Cambridge area. He was largely responsible for the pioneering work on emulsion-based droplet microfluidics technology that are used today for all of these exciting new applications in single cell analysis. So basically, he was a material scientist. So Cliff was a material scientist. So when he looked at this problem, he did something that, say, a biochemist wouldn't think of doing. Of course, a biochemist would, break it down, do mass spec, do bio-ID, blah, 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 look at phosphorylation. They'd collect all this information, and then they'd come up with some sort of pathway to describe what's going on. Cliff took a different approach. He poked the cells. And by poking the cells, he saw the most remarkable thing, which was these P granules looked like they were behaving like liquids. They were sticking onto surfaces, wetting a surface, in this case, a nucleus. This describes the direction of the stress that was applied. And they dripped like a liquid, and they fused like a liquid. And this was the beginning of what has become the field of biomolecular condensates. So anyway, proteins with Prion-like domains also have this characteristic. And by the way, remember that ugly structure I told you? Well, this is what happens to that structure if I filter the network according to whether proteins have Prion-like domains. Suddenly it organized itself into this nice symmetric structure, and it turned out, in fact, these are the proteins of the processing body or pre-body of yeast, which also happens to be a biomolecular condensate. So what is the importance of this? What we thought was that, first of all, that it would be interesting to see if the proteins forming the coat of the coat of the endocytic site and the puncta that are absorbed, or there might be a biomolecular condensate. But taking a step even further, is it possible that the condensate itself could exert some force driving membrane invagination? So to look at this problem, so first of all, this is not a new idea. This is very well known in colloid physics, that when two viscoelastic objects are in contact with each other, the first thing they will do is they will adhere to each other. But because they are liquids, okay, if they start pushing in, and they're tethered in any way at the top, then something is going to have to be distorted, because being viscoelastic liquids, they have to maintain their volume constant. Okay, so and what is involved in all of this, of course, is surface tension. So an emergent property, of course, of any kind of condensate, whether it be a liquid droplet or whatever, is that it has surface tension. We all know about this concept. And in the context of condensates, this has been shown clearly that condensates can do work. Okay, in this particular case, this is now an outstanding paper from now Cliff Brangwen's lab when he moved to Princeton, where he showed that there are two proteins that are probes, that are often used as probes for the organization of the nucleolus. And what's known as the nucleolus has this remarkable substructure in vivo. He was able to reproduce it in vitro, but only under specific conditions. You see, what he showed was that this protein here was for encoded this inner sort of structure, and this protein here encodes for the exterior structure. And what he found was, first of all, these two proteins would form condensates and viscoelastic condensates, but they didn't form this structure until you added RNA. Now, this is the nucleolus, of course, is where ribosomes are synthesized. So this made perfect sense. But what does this mean from a material point of view? Is that the addition of the RNA somehow altered the organization of these two structures that resulted in changes in the relative interfacial tensions of these two structures with each other and with their surrounding, such that I won't go through the details, but the result was basically that under these conditions, both of these objects preferred to be exposed to solvent, but under these conditions, the green one preferred to be exposed to an interface with the other droplet. Okay, so the notion that you could have something do work, like an object undergoing phase separation and doing work by virtue of the interactions with its surface, is something that logical and makes sense. Anyway, so first question was, are endocytic puncta protein condensates? And to do that, we did basically, it's a classic experiment. What you have to show is that there's a clear phase boundary where an object goes from existing to dissolved, okay? And one does this by definition. A phase transition is a transition in which you can periodically and repeatedly and rapidly go from the phase separated to the mixed phase, okay, by repeatedly overcoming the same threshold temperature. All right. This is also important from a biological perspective, because if you were, constantly shocking cells like we're doing here, right? Your question would be, well, what biological process could you be affecting doing that? And shouldn't something like that be irreversible? So if that were the case, you'd start seeing things drifting, the threshold drifting around, and you probably couldn't, there'd be some sort of hysteresis, so you couldn't repeatedly and very rapidly, but in this case within 20 seconds, go from formed to mixed to formed to mixed. But we showed that we could, okay? And we showed that this is just a representation now of the across the population of cells where the transitions occurred, as measured by the number of puncta per cell. And what we see is a sharp biphasic transition at a specific temperature, with of course some variation, but due to biological noise and experimental error. So that's good. Now the other evidence that there might be a condensate forming here was that it's been already known for a fair amount of time that there was a zone around the sites at which endocytosis initiates that excludes ribosomes that are labeled here. These black objects here labeled with immunogold antibodies. What's shown here is you can see here the beginnings of the formation of a invagination in the membrane, and then later, right, a fully invaginated membrane. And actually over here on the left is a mature vesicle, okay? Interesting, in all cases, including the mature vesicle, you have this exclusion zone. And the dimensions here are around 100 nanometers. Okay, so that was interesting. This is also showing up in fluorescence studies in which you can see, looking at different protein probes. This is one that we use very often, SLA1. You heard about this guy. This is the one with all these SH3 domains that Hugo Dion talked about in this interesting study he reported in on the first day. So what they showed was for SLA1 that the relative radius, okay, of this object is around 50 angstroms, so on the same scale that as the object that was seen by EM. And whoops, I'm not getting any. And furthermore, we saw this structure in D-storm imaging. Our measurements were of an object in yeast of about 200 nanometers by 100 nanometers. So, basic question: what was causing? Excuse me. What's causing the exclusion, right? So I'll get into this a little more detail, but the notion is that the insides of these condensates are formed between either intrinsically multivalent or intrinsically disordered domains or multiple structured domains of various valences that form sort of some cross-linked mesh, okay? And that mesh size is going to determine what can get in. So we could probe this with FTSE labeled dextrans of different sizes, 5.8 versus 10.4, and we could perform fluorescence recovery after photo-bleaching experiments where we bleach all the fluorophores in the endocytic puncta and follow how fast dextran re-enters the objects. And what you can see is this happens very rapidly for 5.8 nanometers dextran, but practically doesn't happen at all for 10.4 nanometers dextran. So there's a sifting behavior, which is a hallmark of condensate organization that's interesting because it's been found in a number of different examples that there are both structural and functional implications of this about the interiors of the structures and about their chemistry that are still in the beginning of being explored. We can destroy the, like other condensates, biomolecular condensates, we can non-specifically destroy these things with a compound called 1,6-hexane diol, okay? And when we do that, we don't see any difference for the 5.8, but you can see this at least partial recovery for the 10.4 nanometer dextran. So for some reason, my forward isn't working anymore. Anyway, just quickly, I mean, this has been seen in other cases. You can see here for 3 versus 9 nanometers. You can see the merging of two condensates, the protein condensates LAF1 with dextran here, and it starts disappearing at 6, and by 9 nanometers, dextrans, there's no signal. This has also been sort of seen in the substructure of another condensate formed by prionic proteins, yeast prionic protein sub-35. And the consensus is that the scaling of the mesh size is about 10 nanometers, so nothing bigger than 10 nanometers, and with the right dimensions, they're going to get into these condensates. Okay, some other evidence. As I said, the PLD domains are essential for partitioning of coat proteins into partitioning of coat proteins into endocytic puncta, and we can see that specifically the deletions of the domains for all of the proteins with PLDs results in them ending up diffusely distributed throughout the cell. But not all of the PLDs are essential to function. So what we're measuring here is fluorescent flux into the cell. This uptake of a membrane of a dye, of a fluorescent dye into the cell by endocytosis. And it turns out actually only two of the proteins, coat proteins, had a deleterious effect when you remove PLDs, right? So what that suggests is, but by the way, we tried deleting both and it was lethal. So obviously they're complementing each other in some way that allows endocytosis to work under these conditions. But obviously you do need the PLDs in order for endocytosis to work. Okay. When we looked at the protein interaction network using DHFR PCA, what we saw, let's go to the figure, okay, is we looked just at SLA1, our old friend here, and we see a quite extensive network with proteins involved both in the early endocytic processes as well as in the actin endocytic process. Anyway, when we deleted the PLDs, okay, those interactions were shown in black, and when we deleted the PLD of SLA1, the only thing left were these two interactions here in blue that in fact have nothing to do with the early phase of endocytosis, but are involved in the actin-dependent phase. Okay? And sorry, just as an aside, what we've done is we've rendered these cells such that, just to remind you that we've rendered these cells such that they no longer do actin-dependent endocytosis. So everything we're measuring will be actin-independent. Okay. The other thing we did was we looked at the compositions of the PLDs to see what difference that made. Okay, so what we did was we looked at the overlap between a reporter protein and this particular coat protein, ENT1, which was, sorry, just taking a step back, that was the one that gave the strongest effect, right? So what we looked at here was what happens if we swap in different PLDs into this protein. Is it going to part into this protein? Is it going to partition, and are you still going to see endocytosis? And so what we chose was to swap the NT1 PLDs with that of SLA1, the other one that we know works, right? And with this prionic protein I mentioned earlier, sub-35. In addition to that, Randy Halfman, then in the Lindquist group, came up with these two versions, which have all of the glutamines replaced with asparagine, or vice versa, all of the asparagines replaced with glutamines. Okay, you'll see why this is important over in the next few minutes. So this is what we saw. This is just a summary of the results, and these are the this is right here. These are just the correlation coefficients for overlap between puncta. And what we saw was that for SLA1, you could get perfect association, partitioning of both ENT1 and ENT2 into the endocytic puncta with a SLA1 prion-like domain. Even a human, we took a human ENT homolog, and we replaced its PLD, and it got in, okay? In addition to that, though, the sub-35 with the Q substitutions also got in, okay? Not quite as well, but pretty good. So what's interesting about this is the following, however, that if you did the same substitution and looked at uptake through endocytosis, nothing worked except SLA1. And this is a phenomena the people in the condensate field have come to refer to as the Goldilocks effects. There seems to be some perfect composition and order of amino acids in Prion-Like domains that determine the properties of the condensate, and they have to be just right in order for this to happen. And it gets to some interesting questions about this at the end. But let me take a quick aside to explain some of the details about the organization of condensates and why this is important. So what's come to sort of focus a lot of energy on how intrinsically disordered domains, proteins, domains form condensates is a well-known theory in polymer theory called the associative polymer theory. What it posits is that polymers that form condensates consist of what we call stickers and spacers. And stickers are these multivalent nodes that, for whatever their chemical properties, tend to interact with each other, and spacers are those that define the intervening regions, which can determine their material properties. Okay. And as a consequence, depending on their material properties, you can get anything from a viscous liquid where the network is undulating and re-equilibrating constantly to a solid, okay, in which these interactions are so stable that basically you can't break things up. And these are these are FRAP results again. This is what you would expect for the viscous liquids. The molecules are exchanging very quickly, so you get rapid recovery of fluorescence, but for a solid, you get nothing because nothing can exchange. So I'll just very quickly go through these because I'm running out of time, but in proteins, what's been shown by Tony Hyman's group, and this is also theoretical work from Rohit Papu, is that the basic stickers of prion-like domains are arginines and tyrosines, okay? And depending on the numbers of tyrosines and numbers of arginines, it determines the saturation concentration, the critical concentration at which phase separation occurs. It also determines what types of material properties you have, and particularly in the spacers where when you go from simple amino acids like glutamine to glycine, you see important transitions whereby, for instance, with the Glynn-rich spacers, you end up with proteins that have solid-like behavior, but with glycines, they remain liquid. So now how is that? In addition to that, now I'm really running out of time, so I'll have to go over this quickly. I'll just say that it's not only the numbers of aromatic amino acids, but the patterns that are important. Just illustrate. Just look. Whoops. What? Look down here. Where are we? Sorry. Just look down here. Okay. This is a case in which they have what they call a perfect aromatic, which is the aromatics are spaced equally, versus somewhat patchy to completely patchy. And what you see is something going from these nice perfectly formed liquid condensates to this absolute mess of an aggregate that behaves like a solid. Okay. These are simulations up here, but this is what they look like in real life. Okay. So we can probe the material properties of cytosol and condensates using optical tweezers. I won't go through a lot of detail here to explain this, but the idea is that we oscillate a polystyrene bead with a laser at some given frequency, and we follow the ability of the object to follow the carrier frequency at different frequencies. And what we end up with is this curve. Without going into any detail, what this tells us is that, in fact, when we talk about the viscoelastic properties of material, what we really mean is that it depends on the frequency domain you're working in. To think of it more simply, it has to do with whether you apply a force very slowly or very quickly to something. So think about it like water, where if you put your finger in very slowly, the network of interaction hydrogen bonds among the waters will gently reorganize around your your finger, but of course if you slap it with your hand very quickly and hard, you'll have more like an experience of a solid. So we did that with in fact with the cytosol of the yeast that we were studying, and then we could, basically using some geometrical theory, we could use this to determine the viscoelastic properties of the condensate. We're able then to plug in values for the geometric and material properties of the cytosol and so on and so forth, and plot this energy potential. And what this shows is what we would predict as the invagination, the distance of invagination of the membrane as a function of energy. And you see we could hit a minimum somewhere here around 40, and in fact the minimum for the actual total trajectory of invagination in cells is about 70 nanometers, which in fact it fits within the range of energies that we'd expect. And so just sort of in a mechanical analogy of what we think is happening as a summary is that as the condensate grows on the surface of the membrane, okay, it does two things. First of all, it's adhering to the cytosol, but at the same time, the cytosol is pushing back because it's elastic, okay? But as it grows, the surface energy, obviously the surface area increases, the surface energy, the surface tension increases, right? And at the same time, the adhesion increases. Now, because this force, okay, the force that's being generated by this is being applied symmetrical, and because this is the condensate is a viscoelastic fluid, something's got to give, and the only thing that can give is the membrane, and it gets sucked in, okay? In addition to that, by getting sucked in, that increases the surface area, and you get effectively a positive feedback loop leading to more and more and more invagination. Okay, so that's basically the story. Okay, and I'm going to skip over some of this, which is some of the implications of this in disease, just to wrap up with mentioning some of the things that we're doing now, which is that we have sufficient evidence now that all of these different types of processes in the cell, and including viral maturation, involve formation of condensates, okay, and that in some way are driving these morphogenic forces on membranes in the cell. This is also being observed in the nucleus by other groups, where now it seems like virtually everything about the organization of chromatin and nuclear subfunctionalization seems to have something to do with protein condensates. Moitza Yussa's work with Brenda Andrews, and she talked about yesterday, and I'm looking forward to hearing more about that and the work she's doing in Ryerson. So I'm going to end with that, and I just want to actually, before I go to this slide, I'll end with this slide and just say one thing. These are all the folks I worked with and did the optical trapping experiments with Alan Ehrlicher and Adam Hendricks over at McGill Bioengineering, where I'm an adjunct, and Paul Francois in the physics department at McGill. We also worked with Rohit Papu, who is sort of the reigning guru of protein condensate theory in the world today. So it's been a pleasure and honor and really amazing to work with him. But the person I really want to talk about is Louis-Philippe Bergeron-Sandoval, who was the student who entirely led this project. And so there are two remarkable things about him. One is the number of syllables in his name. And the second thing is that he proved an important point that I've learned through my career, which is that when you've got extremely talented students, you got to let them run with it and do whatever they're going to do, because you're going to learn fantastic things. And I've learned more about the organization of matter in cells in the last 5 years as a result of working with him and my colleagues here than I ever have in my life. So with that, I'd like to thank you all for hanging in there till the end of the talk, and I look forward to any discussions.
Thank you so much. Imagine the hundreds of people that are on Zoom are clapping at the same time. This was really, really informative and interesting. novel for most of us. Did you skip over the slide about the relationship with network biology?
Yes, I'm sorry. I thought this up because you put the challenge to me. Sorry, I just want to tell everyone this story. I mean, I happened to talk to Anne-Roxandra on the first day, and I told her, you know, I'm really honored to be at this meeting, but I don't know why you invited me because I really haven't been doing any interesting network work for a while. And she said, she wrote me a nice note later and said, oh, no, I don't want to hear about networks. I want to hear about what you're doing now. And so I had to adjust my talk. That's why it ended up being sort of a two hybrid approach. And sorry, that was a terrible joke. But that's why I put this talk together the way it is. And so what she asked me was, what do you think are some of the challenges that network biology may be geared to take on, and may in fact be privileged to be able to make special contributions to this field? And so I'm just putting this up in case it might inspire some discussion or questions.
Thank you very much. Let's start with one question we have in the Q&A from Augustine Cruz. Right, thank you for such a wonderful talk. I learned a lot. So did I, and so did most of us, Stephen. I have a question about the role of ionic forces around these structures. Is the formation of these puncta structures related to the change in ionic force of the environment? Is there an ionic movement along with the formation in these structures?
That's a great question. And you know, I alluded earlier, not alluded to, I explicitly talked about, you know, how we discovered this whole process by looking at something completely different. We looked at phosphorylation. Charge distribution is absolutely critical to the way that condensates behave. And we can imagine that there are both internal and surface properties that are driven by the structures, by the distributions of molecules and associated salts, right? One thing I think if you're looking for something interesting to look at in the future in this area is that I believe that the most interesting relationships we're going to find with respect to condensates and cellular structure generally involve heavy metals, like zinc and copper and iron, okay? Just as a hint, okay, something we've been interested in and it had some results that were exciting was that it seems persister cells, both bacterial persisters and cancer persisters, these are cells, of course, that are resistant to whatever antibiotics in the case of bacteria and chemotherapeutic agents in the case of cancer cells, share one thing in common. They both exist in a more what's called glassy state than the rest of the population. So there's something happens, or there's something about a subpopulation of cells that have these particular properties that are interesting. And we were looking at one aspect of this, and what we're seeing is that when you drive cells into a particular stationary state by starving them, hundreds and hundreds and hundreds of species of condensates form. So.
Wow.
Yeah.
That's crazy. So, if any of the panelists has a question, oh yeah, Pascal has one, please.
Fumble with my microphone first. That was beautiful, Stephen. I heard you talk about these condensates like for the first time like 3, 4, 5 years ago. I was already fascinated. It's fantastic.
In Toronto, I remember.
Exactly, yes, and it's fantastic to see how this develops. So, what do you estimate? I mean, when the spacing of amino acids is so important for the function of these, they should be easily identifiable. What proportion in a proteome of proteins do have these condensate forming properties? Do they stick out in terms of how they're interacting in the network? And how does it's 3 questions in one. And how does it change how we need to model signaling and on all these biomolecular pathways if we go to ODE-based modeling, and how do we account for this?
It's a great question, and the answer is I don't know. That's the short answer. The long answer is that it's going to be a lot. We know that of 2 principles, as I mentioned, and I think there's at least one other principles that drive protein phase separation. One that you mentioned, of course, is intrinsic disorder, of which we know 30% of the proteome is to begin with. Okay, so there's a potential there. multi-domain proteins like SH3 domains, like you heard from Hugo Dion in the first talk, right? The discoveries of Mike Rosen about the importance of multi-domain structures in forming condensates. Well, you know, in Betazoa, how many of those are there in the proteome, right? It's frightening to even think about it. So it's going to be a lot.
And thank you. I think you've answered all of Pascal's questions. Yes?
I think it's frozen, huh? No.
No, not frozen.
Oh, that's muted. Pascal. But along those lines, there is a comment in the Q&A from Miguel Janowski. I was happy to see something about biological condensates here at this conference. This is, this is becoming increasingly a focus in circadian biology as well. There seems to be a time of day change in these condensates, and a lot of intrinsically disordered proteins are involved in the molecular clock. I will certainly let others know about the work you highlighted here today. Thank you.
Thank you.
So I think she's commenting on something that probably we all felt listening to you, is how these condensates and generally the macro properties at the scale beyond proteins and interactions, single molecules, even multimolecular complex, have these physical properties that really will influence all of cellular biology, signaling, movement, and how, as network biologists, can we incorporate this in the models, whether it's the signaling networks, the genetic networks, etc. How do we take this and take advantage, both to learn from it and to learn about it?
Yeah, I learned a couple of things from this meeting that are pertinent and really surprised me. One I could say is, well, I think Suzanne Goudreau made a really pointed comment that captures, I think, the state of the network field, which is that what we're learning is that you need to know two things. You need to know fine detail, and you need to know global things about network behavior. And these are the challenges that, you know, that remain, but which I've seen at this meeting that in 10 years have made fantastic progress. It's really exciting. And I think that those are going to be relevant to condensates as well. Marlinka Zitnik had a result that blew me away yesterday. I just wanted to share it with you too, because it points to this. What she said was that they tested something, 76 out of 77 drugs, and it turned out that their actions had nothing to do with a specific target, but to the organization of the network. And that just blew me away. And it's been something I've been thinking about for 2 decades. Is I didn't understand in our own experiments how we could see these gestalts where you would have, you know, like just a few reporters, and you would, and yet you would pick up mechanistic information that was deeply insightful. But what I pointed to also in the field is how important it is to have two things. So I think nowadays what we can do is really simplify experiments using, you know, fewer probes and doing you know, networks in batch, like these interactome in a tube approaches that, you know, Ben Weller talked about, Fritz Roth developed. And but we can only do that because of all the comprehensive work that's been done, right? Because we need to actually use this information. We need the details of the networks.
Thank you very much. Those were, I think, fantastic closing remarks about the interaction of what we've learned this week in the conference and how it will take us to the future. And we are excited to see this feature develop and include the physics, the condensates. Thank you so much, Stephen.
Thank you.